So my name's Sean Burns, I'm Medical Director at Intellia, and I'd like to share with you a little bit today about our company, about why we're excited about CRISPR and gene editing approaches for rare disease, and also some of the challenges that we're facing to bring this to the clinic. So I am an endocrinologist by training. I still see patients, a fair number of which have metabolic disease, but certainly not rare, mostly type 2 diabetes, which affects about a third or so of individuals, adults in the US, either in the overt form or pre-diabetics. So Intellia Therapeutics is a company that was founded about 2 years ago, opened its labs in early 2015, and this is a company that's entirely focused on bringing gene editing to the clinic for serious disease, and most of those diseases tend to be rare genetic disorders. The company had a successful IPO earlier this year, which has really helped to galvanize a lot of the programs that I can talk to you about today. We're located in Cambridge, Massachusetts in the United States. I joined in July of last year. I was employee number 24, and we're currently up to about 100 employees. So we're growing pretty rapidly. And again, a lot of that is because of the excitement and the interest that's allowed us to grow so quickly. We have intellectual property rights to CRISPR estate from Jennifer Doudna's group at UC Berkeley, and also tied into Emmanuel Charpentier's intellectual property as well. And we are, again, we're a very small company, and we're very eager to form collaborations that will help us get into the clinic quickly and allow us to get therapies to patients as expeditiously as possible. So we have two major collaborations underway right now. First is with Novartis, and this really focuses on ex vivo applications, which we've talked a little bit about ex vivo versus in vivo, and I can talk a little bit more about that. That's focused primarily on CAR T cells for immuno-oncology applications, as well as on HSC-targeted therapies. And then more recently, earlier this year, we announced a partnership with Regeneron, and this is really focused on bringing CRISPR to bear for diseases of the liver, which I can also mention a little bit more about. So fortunately, a number of excellent presentations before mine have really gone through how CRISPR works. I guess this slide is really meant to show you how we think about CRISPR and its role and why we're so excited about it. So really never before have we had a way of approaching genes and making genetic modifications to our human genome with efficiency and precision in a way that CRISPR enables. And so what we really start to think about is that this tool allows us to perform surgery at the molecular or DNA level to change a predilection for a disease or to stop a disease that is already underway by changing really at the source, changing the DNA, the code itself. So again, I'm not going to go into the components here that exactly how it works, but I would just say that one of the advantages of this technology versus some of the predecessor gene editing technologies is that the vast majority of this system remains constant between different targets, and so very much different from zinc finger nucleases or TALENs, which were the sort of the precursor gene editing tools, as well as meganucleases. So the nuclease, which is the protein component here in gray, is the same, and so actually as Dr. Fulvio Movilio was mentioning in the webinar, this is sort of a universal scissors, I think is the term that he used, or in this case, scalpel, if we're thinking of the surgery metaphor. So having that constant protein enables us to characterize and understand the system in a way that is would be very difficult to do if the protein is changing. We can understand characteristics such as the pharmacokinetics of the protein, how it's degraded, its half-life, immunogenicity. All of these things are relatively constant for this system. What changes is the RNA sequence, the guide, and that there's a 20 base guide in most systems that really determines where this scissors or scalpel goes to cut within the genome. And that's, of course, the key component that really enables us the flexibility to target different genes. So much like traditional surgery, the reason that the system is so powerful and that it works well is that the body heals. And so really, even at a cellular level, as we've touched on earlier, after you make a cut with CRISPR or actually any gene editing technology, the outcome of that cut is determined by the cells. And so if in the most sort of the default case, which has been discussed before today, sorry, the microphone's coming apart on me, through a process called non-homologous end joining, which is shown on the left, this process is referred to as error-prone. That's not exactly precise. It's actually, the DNA most likely heals itself very, very efficiently, probably 99% of the time. But compared to some of the other, relative to the other ways in which a cell or DNA can heal itself, it is more error-prone. So if 1 out of 100 times it introduces either a small insertion or deletion, and if it can continue to be cut, if it repairs itself perfectly well, eventually those genes or that sequence will accumulate deletions at that location or insertions. And so that approach is very useful for introducing what we call frame shift mutations, which cause loss of function of the targeted protein. And then there are, in the middle and on the right side here, are 2 other repair pathways which involve providing a template to bias the outcome of the repair. And so in the middle, using a single-stranded DNA template, we can use a process that homology-directed repair to change single-point mutations. And then on the right-hand side, we can actually imagine doing full insertions at targeted loci using homologous recombination. And there are actually other flavors of corrections that we could talk about as well, including actually inserting pieces of DNA in targeted loci through other repair pathways. But we're very interested in taking the full advantage of the CRISPR-Cas9 system, which really requires that we solve efficiently biasing towards these outcomes. And so with that in mind, our company has interest in, has programs that align with each class of edit. And so we call these our sentinel programs. So for the knockout approach, we have a program focusing on hereditary amyloidosis and a gene that's causative there called transthyretin, or TTR gene. Knocking that gene out should have a therapeutic impact in those patients. Similarly, for repair of a single point mutation, we're focusing on a condition called alpha-1 antitrypsin deficiency, of which many in this room are familiar. One single allele there is really causative for the disease, and if we could change that one allele back to the wild type, we would essentially be able to cure a lot of disease in those patients. And then on the right-hand side, again, sort of probably the highest bar in terms of outcome would be to have an insertion or correction of a large portion of DNA. And many, if not most, genetic disorders would fall into that category, most disorders being recessive with many ways in which a gene can be mutated and cause loss of function. And in that case, we're focusing on a number of inborn errors of metabolism. Again, thinking about how we can solve these problems to not only for the diseases that were listed here, but also as a way of opening the door to other diseases of a similar nature. So I think this has been shown a little bit before today as well, but just to sort of put this into context and how it compares with gene therapy. So gene therapy, there's some indications where gene therapy is probably the better way of going if you can achieve delivery and expression level that's therapeutic. And there are certainly some indications where gene editing would be preferable. So just sort of broken down here a few categories in which they're different. And again, this has been mentioned to some degree before, but gene therapy involves inserting a healthy copy of a gene into a cell that has a defective copy. That's sort of in contrast with gene editing, where we're talking about correcting the mutation within the normal cellular context, so not providing any additional genetic material to the cell. Usually with gene therapy, we're talking about delivering with a virus, and the reason for that is because we want persistent expression of the transgene, the gene that we're putting in. This is in contrast to gene editing, where we actually want transient expression of the CRISPR system. And we want the gene editing to occur, and then for the CRISPR system to go away or to get degraded. And so as you think about delivery, there's a big difference in terms of which ones would allow you to have transient versus permanent effects. With gene therapy, we talk about lentivirus potentially integrating within the human genome in a semi-random fashion, or AAV maintaining as what's called an episome outside the genome but still within the nucleus. That's very different from gene editing, where we're actually going in and correcting the point mutations within the human genome. And again, as I mentioned before, the expression that you'd want with gene editing is transient versus permanent. And one of the components here is that with a transgene approach driven by a vector and a promoter and sort of that sort of a system, usually the expression that you get from that is higher than the normal expression of the gene, and oftentimes it's not exactly physiologic, so it may be higher and less variant or less dynamic. Gene editing affords the possibility of actually maintaining the normal expression system for the gene of interest. So if you correct the point mutation but don't cause any, don't affect the regulatory aspects of the promoter or the enhancers or everything else that goes with the gene, then that expression should be maintained at the normal physiologic level. So really, the CRISPR system is so efficient that in some ways the challenge of delivering, of getting CRISPR into the clinic and treating patients comes down to challenges of delivery. And so this is something that has been brought up before today as well, but we are focusing on two general categories of delivering CRISPR, some on the ex vivo side and then others in the in vivo side. The ex vivo side, as was mentioned earlier today, there are tricks to get CRISPR into, for example, hematopoietic stem cells, and you can either insert protein or actually insert RNA and other approaches that are relatively well understood and more straightforward. I would say that the bigger challenge here is on the in vivo side and figuring out ways of injecting something into the human body and getting that to the cells that it needs to get to, and potentially surviving the immune system, surviving or getting to the cells and actually having the effect that you want, and then actually going away for CRISPR. And so in terms of in vivo delivery approaches, we're focusing on developing two of these, and thankfully Shoban has already introduced AAV, and others have as well mentioned this. We're also interested in another platform called lipid nanoparticles, and so this is another strategy for delivering nucleic acid therapy to cells. This is not a new approach. This has been around for decades and has been used successfully in patients for delivering siRNA therapies, or RNA interference approaches. And the tropism for each of these approaches is quite different. So by that I mean where these modalities take you within the body. So lipid nanoparticles have been primarily developed to target the liver, and there are approaches to expand on that, but I would say for now that's where most of the effort has been placed. AAV, on the other hand, can go is much more flexible, so you can actually go to many other tissues by changing the protein that caps it, or the sort of the outside surface of the virus, and those are referred to as different serotypes that get you to different organs and tissues within the body. On the lipid nanoparticle side, it's quite large, and so you can actually fit quite a bit of genetic material in there, whereas the AAV is somewhat limited in size, and so you have to figure out tricks to fit the CRISPR system, which is actually itself quite large, into that. Immunogenicity is quite low for lipid nanoparticles, but AAV, because it's prevalent naturally in the environment, a lot of us already have pre-existing immunity to the AAV, and so we have to develop ways of moving past that. And also, as we think about repeat dosing, AAV is tough to imagine a repeat dose systemically that would not be subject to the pre-existing immunity from the first dose. The duration of action of lipid nanoparticles also is transient, which lends itself very well to CRISPR. AAV, because generally it's long-term, we have to figure out tricks to inactivate the CRISPR system after it's done its job within the cells. So there's a lot of effort there, both from industry and academia, to develop these approaches. And then manufacturing, which again Shoban mentioned that Spark has really spent a lot of time working out the kinks in the system to make AAV in-house, but it's a very tricky system to get to work and to do at scale and to produce high-quality virus that would be enough for systemic administration, let alone to the smaller volumes of the eye. So lipid nanoparticles, in contrast, are much easier to scale, chemically defined. There's no bioreactors involved. So for delivering to everywhere we want to get to, at some point, I think AAV will be required, or something similar to that. But for getting to take advantage of CRISPR and to move to the clinic expediently, we're focusing right now on using lipid nanoparticles as our first generation in vivo product. So I wanted to share with you a case example here of lipid nanoparticle delivered CRISPR in mice, and this is to target the same gene that I mentioned before, TTR, for amyloidosis, but these are in wild-type normal mice. So if we look at just taking mouse hepatocytes in a dish, this is on the left-hand side here, we can increase the dose of these lipid nanoparticles containing the CRISPR system and actually see editing percentages of upwards of about 75% or so just in a dish without any selection of the cells. If we take those same lipid nanoparticles containing the same guides and put them into the tail vein of a mouse, we can achieve editing in the liver of the mouse of about 50% or so, and that's actually counting both the hepatocytes as well as the other cells in the system that are not taking up lipid nanoparticles as efficiently. And probably more importantly than that is what do we see when we actually achieve that editing, and we can see a knockdown that's dose dependent in terms of the serum levels of TTR in these animals. So we're actually, which is really what we care about in terms of treating this disease. Can we get rid of this protein in circulation? And this is early data, this is proof of concept, but this is certainly not, there's room for optimization here for sure, where we can actually achieve a therapeutically meaningful percent editing where we actually see TTR levels go away, and so we're actively working in that. So, I'm short time-wise, but I did want to just mention some of the other conditions we're working on. So I mentioned TTR, alpha-1 antitrypsin deficiency, which ultimately we're hoping to have an efficient repair of the single point mutation. There are some patients who would benefit in the near term from a knockout of that gene, those patients who have a liver phenotype. Hepatitis B virus has a latent form which involves a DNA, almost a mini chromosome of sorts within the infected hepatocytes, so we may be able to use CRISPR to clear that. And then there are a host of inborn errors of metabolism, some of which would be amenable to knockout approaches, others would be amenable to the higher order classes of edits, including single point mutation correction and insertion of gene material. And then ex vivo, as I mentioned before, CAR T cells and HSCs. So just in terms of challenges, we think about broadly about technical and regulatory challenges. So again, delivery is really key. How do we get to the CRISPR system to where it needs to be to achieve therapeutic benefit? And can we get that editing percentage up high enough where, whether it's within a particular cell, can we efficiently knock out or edit both copies, both alleles, and then in a whole organ or tissue level, can we achieve enough editing within an organ to actually achieve therapeutic benefit? These are certainly challenges that are high on our mind to accomplish. And then, as has been mentioned before, we have a high bar in terms of safety here. There is always the potential for a system that's as powerful as this to cause problems if it's not specific in where it's cutting. And so we've spent quite a bit of time, and we continue to spend time mapping the off-target events that can occur from the system and figuring out ways of minimizing those off-targets. And we can talk more about that. And then, of course, because this is a bacterial enzyme, there is the concern for immunogenicity, and we're looking into ways to minimize that or to mitigate that from a therapeutic perspective. On the regulatory side, I won't read through the list of things here, but there's certainly quite a lot that's I would say, perhaps not unique compared to gene therapy, but certainly issues that are distinctive compared to traditional pharmacologic therapies. And one thing that is distinct from gene therapy, if we are going after point mutations, this patient populations that we're treating may actually be smaller than the overall disease patient population because we're actually developing personalized medicines that are sequence specific. And so that adds another layer of complexity to regulatory issues. This idea of designer babies is certainly out there, and we have to sort of try to keep the focus on what we're talking about here, which is targeting genes from a therapeutic perspective. And more recently, another great publication, The Hollywood Reporter, mentioned that Jennifer Lopez has a new drama coming out about hacking people's DNA. So, in that context, we actually really would appreciate, the folks from Eurordis trying to keep the message on target here, that people say, oh, that CRISPR thing sounds really scary. Say, well, there's pluses and minuses of powerful technology, but I think just to kind of keep an open mind is really the key message here. So, and really we have to solve these problems because ultimately what we're working on here is to really have a strong impact on patients and patients' families who live with these diseases. So with that, I'd like to thank you for your attention and be happy to take questions.
Okay, so there's just a handful of items that I wanted to cover today. The first is to reflect on today's presentations. I then wanted to talk a little about Vertex's commitment to people living with cystic fibrosis and talk about how gene editing will play a role in Vertex's mission to help people living with cystic fibrosis. And then finally I wanted to talk about how Vertex's commitment to the treatment of rare diseases is moving into conditions beyond cystic fibrosis. So in terms of today's presentations, I think the workshop's been really well structured. Thank you, Leslie. I think we've had some excellent speakers, and we've covered a whole host of topics from the science and CRISPR as a technology and how exciting it is to have this technology now, the clinical reality of the conditions that we're trying to treat, the ethics and the legal environment and the challenges that poses. And obviously we've heard a little bit about what biotech companies are trying to do with the technology and the challenges we face. And I think by and large Vertex is aligned with what we've heard today. I think certainly we understand that CRISPR and gene editing is its infancy, and the first steps we take with this new technology have to be careful and considered. Just to pick up on one point on the ethics, I think Anne Lean's presentation discussed the ethics really nicely. Vertex and our collaborator, CRISPR Therapeutics, we have a specific position on, we're only looking right now at developing therapeutics for somatic cell gene editing, so the treatment of diseases. So right now, we don't think that germline editing is appropriate right now, but on a personal level, I think it's right we have the scientific debate for sure. Okay, so moving on to our commitment to people living with cystic fibrosis. So it may be that some of you have heard of Vertex before and the work we've done developing treatments for people with cystic fibrosis. If you haven't, I hope you leave the session today remembering that, and also that we're trying to move into other rare diseases. But in essence, I think all of us at Vertex share a vision that we can make a positive impact over time on the lives of all people living with cystic fibrosis. So cystic fibrosis is a rare genetic disease. It's autosomal recessive. It affects many organs in the body. Primarily, it's associated with being a lung disease or a respiratory disease, and patients over time lose respiratory function. I think there have been big improvements over the last 20 or 30 years of how the disease is handled, both from a medicine's perspective, but also through other treatments such as physiotherapy. But the fact is that people living with cystic fibrosis are still have a reduced quality of life, and they're dying much younger than we would like them to do. In terms of where we find cystic fibrosis, so it's a disease that has Celtic origins. So we find mutations in the CFTR gene in people of Celtic origin, which mainly puts the disease in North America, Europe, and Australia. Vertex is working with the cystic fibrosis community around the world to try and develop treatments for this condition. Okay, so I guess of the three presentations we'll have from biotech companies, mine will be slightly different from Sean's and Siobhan's, in the sense that Vertex are not solely focused on developing treatments either based on gene therapy or gene editing technologies. So we already have 2 marketed products for the treatment of cystic fibrosis. I'm not going to mention the names, this is not a commercial session. But yeah, these medicines they're small molecules, they're tablets, they're based on chemistry, traditional medicines, for lack of a better way of putting it. But they're still innovative medicines. So these medicines have been developed to match up specifically to people with specific mutations in the cystic fibrosis gene. We're also looking at developing different combinations, again, of small molecules to develop new tablets that will be able to target people with slightly different mutations in the cystic fibrosis gene. But we realized that small molecules aren't going to be enough to help us meet our mission of helping everyone with cystic fibrosis. So we understand we need to move into the field of advanced therapies. So we have 2 research programs at the moment. The first is in the field of mRNA therapies. So mRNA therapies, they're a bit like gene therapy. They're not quite the same. It's still introducing nucleic acid into the cells, but the effect is deliberately temporary rather than permanent. So we know that mRNA therapies will have to be given repetitively, probably not daily, but maybe fortnightly or monthly, something of that nature. And then we're also, being here, as you can expect, interested in gene editing therapies for the treatment of cystic fibrosis. And specifically, we're interested in the use of the CRISPR-Cas9 technology to do this. So you can perhaps start to see that gene editing is viewed within Vertex as part of a jigsaw, and we can use that to put together the whole puzzle, and the whole puzzle is finding a treatment for everyone with cystic fibrosis. So I go into that in a little bit more detail on this slide. So you may recall from one of the earlier slides that there are 75,000 people globally, approximately, with cystic fibrosis. So on the slide, every kind of person stands for around about 1,000 people living with cystic fibrosis. So I'm going to do a series of builds to show you how the kind of jigsaw comes together. So with our first product, currently we can help about 4,000 people with cystic fibrosis. And that's because this product is matched up very specifically to mutations that these people have in the cystic fibrosis gene. With our second product, we can treat some more, and that's great, obviously. And then we find that for the kind of the green people, as it were, we're conducting more clinical trials with the 2 products we already have approved in slightly different mutations. And we think there's potential for these people to be helped by these 2 medicines. This is mainly in pediatric patients, so very young children. Obviously, we start the trials in adults and adolescents, and then work our way through to younger patients. And all of this is obviously planned and coordinated with the regulators. So here in Europe, particularly with the Pediatric Committee at the European Medicines Agency. As I said on the earlier slide, we're working on the future combinations. We have many different combinations that we have in our early pipeline. Again, to treat some of the rarer mutations that we find. But there's still a group of patients where we know we're going to have to rely on these advanced therapies. And these are patients where perhaps they don't have any CFTR protein at all being expressed. Perhaps their mutation means that there's a stop codon early on in the gene, which means that none of the protein is being made. And it's really here that we'll be looking to use the gene editing technology initially to provide these patients a treatment. So I think this is how we see the jigsaw coming together at Vertex. If we turn now to perhaps the science fiction, to put it as Dominic described it earlier, I guess one day, many years from now, and I think we are talking over 15 years for sure, it could be that we will be able to help all people living with cystic fibrosis via a gene editing technique. It's going to be extremely challenging. I think Sean spoke to some of the issues with the in vivo delivery. Right now, there's no clear answer there, but I think CRISPR-Cas9 does have this unique ability to search out and select certain parts of the genome. So I think the long-term goal has to be that we might be able to use this technology to treat all patients with CF. But to manage expectation, this isn't going to happen anytime soon. Okay, so that's where we are with what we're doing with cystic fibrosis. So in terms of the gene editing, so let's talk about that a little bit more. So Vertex, around about this time last year, started a collaboration with a company called CRISPR Therapeutics. So CRISPR Therapeutics are a Swiss company. I think most of their operations at the moment are in Massachusetts in the US. And as the name describes, they are focused on using CRISPR-Cas9 technology, much like Intellia, to develop therapeutics. Currently, we have two main research programs that we're collaborating with CRISPR Therapeutics on. The first is in cystic fibrosis, and the second is in the field of hemoglobinopathies, which we heard about a little bit earlier. So this is where the hemoglobin in the red blood cells is formed incorrectly, so you get sickling and other problems. So I'd like to kind of talk about that in a little bit more detail. So I think some of this general process was discussed earlier, and I think the hemoglobinopathies program that we have with CRISPR is probably, well, it's certainly more advanced than the program we have in cystic fibrosis. The main reason for that is that the plan that we have is to do the gene editing ex vivo. So the kind of the schematic here is laid out, and you can see that this is likely to be more of a process rather than a kind of a traditional medicine. So we're going to have the situation where a patient is brought into the hospital, they'll have blood cells removed, the blood cells will be sorted to obtain blood stem cells. The stem cells would then be treated with the CRISPR-Cas9 in a test tube completely away from the patient, and then the corrected cells would be returned and reinfused to the patient. So in this way, it's going to be more of a process than the patient turning up at the doctor and going to the pharmacy and getting a set of tablets. It's going to be quite a complicated procedure. So I think, as well as being a bit more easy to deliver the CRISPR-Cas9 technology, I think also starting with hemoglobinopathies and doing the editing ex vivo, we feel is kind of the right balance to the benefits and risks of using this technology. So I think you've seen this quite a lot, that by focusing on one organ first, so the blood or the eye, as we've heard about earlier, we're kind of limiting the risk a little bit until we kind of really understand the technology and how it can be used and the optimal way of using it. So we certainly subscribe to those kind of more cautious approaches at this point. Okay, so I think that's really where I want to get to in terms of where we see gene editing fitting into our pipeline. I think the only other reflections and thing that I want to say today is that what struck me the most is that how much we need to work together. So obviously the scientists that we heard about from this morning, and the clinicians, obviously their input is key. We need to understand the ethical and legal framework. Obviously, I think there's a definite place for the biotech industry in bringing this technology into therapeutic products. And obviously the input from your questions and comments today as patients and caregivers and representatives of patients and caregivers is key. So I think the kind of the takeaway message for us back at Vertex is that we're all in the same boat in terms of developing treatments for rare diseases. I'm hoping that it's not this sort of boat. I understand that this is kind of a risky slide for British person to be showing in the current climate, but I think though sometimes it's better to laugh about these things than get too depressed about it. I think this is probably a better book to focus on. So definitely Oxford since you asked. So yeah, I think we're all in the same boat here. I think there's a place for biotech in this. We want to be part of this. We need to be part of this. But I think that partnership is going to be key to realising the promise of gene editing technology and bringing therapies for patients.
5 minutes that are left for this session. What I would like to do is just to show you that because genome editing is a new technology, it raises a lot of issues, not only on the ethical ground, clinical field or scientific field, but it also raises issues for the regulators. And of course, today I will not go in details into the regulation of advanced therapies, but what I would like to do is just to point two very specific questions. The first one is the classification of gene editing, and the second one deals with orphan similarity, because these issues are discussed at the CAT, and I know that Chris and Michele are taking part to these discussions. And to my knowledge, there is, at the moment, no real answers to these two very burning questions. So if we look at the present regulation, you know that from now 8 years, there is a new regulation for what is called advanced therapy medicinal products. And there are 4 different types of ATMPs: the gene therapy medicinal products, the cell therapy medicinal products, tissue engineered products, and combined ATMPs. So one of the question is whether or not genome editing may belong to the class of gene therapy medicinal product. And I think it's really important to read carefully the regulation, because the regulation is a kind of legislation, and each word is very important. So gene therapy medicinal product means a biological medicinal product, meaning that any medicinal product that is not from biologic origin cannot be a gene therapy medicinal product. And this product has the following characteristics. The first one is that it contains an active substance which contains or consists of a recombinant nucleic acid used in or administered to human beings with a view to regulating, repairing, replacing, adding or deleting a genetic sequence. And also, the second characteristic is that its therapeutic, prophylactic or diagnostic effect relates directly to the recombinant nucleic acid sequence it contains, or to the product of genetic expression of this sequence. So, gene therapy is a very specific class of product, and for example, the regulation also says that gene therapy medicinal products shall not include vaccines against infectious disease. For example, if you have a recombinant vector that is aimed at treating an infectious disease, it is not considered as a gene therapy medicinal product. So the question is whether or not gene editing belongs to this class of products. And of course, the classification is in the remit of the CAT, and the Committee for Advanced Therapies has to classify the product. This is a very simple procedure at the CAT. But then, when the question comes to the CAT has to classify the product. And so we now know from all the presentation that we have today that if we look at an initial genome, normal genome, what we can do by gene editing is either to modify the coding sequence of a gene with addition or deletion. You can also modify the non-coding sequence, modify the promoter or the polyadenylation of site of a gene, or even modify some regulatory sequences that are outside the gene, and for example, just to modify the splice site that will make the possible what has been shown this morning regarding the Duchenne muscular dystrophy. So there are different ways of playing with the genome by using gene editing technology. So, as it has already been said, you can consider two types of gene editing in the same way that you can consider two types of gene therapy. Ex vivo, by modification of cells outside the body, and in vivo, by directly modifying the cells inside the body. So if we look at this very simple table, and if we look at ex vivo gene editing, for example, the first column, is the product that you use to modify a cell a biological product? Yes. Usually the final product are the modified cells that you will inject into the body. So the product is obviously a biological product. That the product contains or consists of a recombinant nucleic acid, the answer is yes, because you have modified the genome of the cells. Then of course the product, meaning the cells that have been modified, they do contain a recombinant nucleic acid. The question is whether the therapeutic effect relates directly to the recombinant nucleic acid sequence. The answer may be yes or no. If you modify the genome of the cells or the gene that is expressed in, to treat the disease, then of course the therapeutic effect relates directly to the recombinant nucleic acid that is in the cells. However, if you look at this example, and this is what Anarita told to us this morning, that if you want to regulate expression of the globin genes, and for example, you know that in the fetus, you have expression of the gamma gene of the gamma globin gene, and from during after birth, of course, there is a modification of different regulatory factors. And the consequence of this modification is that the expression is now the beta globin chain that is expressed. And if you look at these proteins, SOX6, for example, or BCL11A, the expression of these two proteins, which are not expressed in the fetus, but are expressed in the adult, will shift the expression from the gamma chain to the beta chain. So if you can, by gene editing, modify the gene for these two proteins, and if you can knock down the expression of these two proteins, obviously, and this is exactly what Anarita is willing to do, if you can modify these two sequences, then you will modify the expression of the globin chains. And then the therapeutic sequence can be considered at the beta globin, the gamma globin. So the expression of gamma globin is the way the therapeutic effect will be brought to the cells. So the therapeutic sequence can be considered as the gamma globin chain. Then, if you have knocked down the SOX6 gene, is the modification directly related or not to the expression of the therapeutic sequence? It is a question we had at the CAT, and it was really long discussion to know whether we could consider that this was, the modification was directly related to the expression of the therapeutic sequence. So this is just to show that in some cases it is really difficult to say whether or not all the definition of the gene therapy medicinal product are fulfilled by gene editing. And it is exactly the same for in vivo gene editing. If the product, the biologic, if you use, for example, an AAV vector to bring into the cell the CRISPR-Cas9 or any protein that will be able to edit the genome inside the body, then of course, usually, yes, the product is biologic. That the product contain or consists of recombinant nucleic acid? The answer is yes or no, because you know that a recombinant DNA is a molecule that is formed obviously that is formed of a sequence, a polynucleotide sequence. And if you look at different kind of proteins nucleases that can be used to modify the genome. If you look, for example, at ring finger nucleases or talent nucleases, you can have these proteins because ring finger nucleases and talent are proteins. They are not. You don't need any nucleotide sequence to make a whole or just to modify the genome if you use ring finger nucleases or talent nucleases. This is not the case for CRISPR-Cas9 because for CRISPR-Cas9 to target the CRISPR protein to the genome, you need to have what is called the guide sequence, which is made of nucleotides. But if you just want to knock down a gene using ring finger nucleases or talent nucleases, you don't need to have a nucleotide sequence. And this is what is shown on this cartoon. What you can do is what has been said just before, that you may use AAV vectors to carry out the protein or the gene encoding the nuclease or the gene that will modify the genome to edit the genome and to modify its sequence. But if you just want to stop the expression of the gene, you can use what is called a viral-like particle. This is a particle. The external part of the particle is like a virus, but inside this particle you just have protein, you don't have any nucleic acid. And these viral-like particles will be able, if they carry ring finger nucleases or talent nucleases, they will be able to modify the genome, but you don't have any nucleic acid sequence inside the medicinal product. So this is why it's sometimes difficult to make the classification of gene editing and to know whether gene editing medicinal product have to be classified as gene therapy product or not. So, of course, if you don't have any nucleic acid sequence, obviously the therapeutic effect is not related to the expression of the nucleotide sequence because you don't have a nucleotide sequence in the product. So this is just to make clear to you that if gene editing is usually classified as the gene therapy medicinal product, it is not always the case. Then what is the gene editing medicinal product? In that case, this product could be a biologics, like any other biologic. But then, of course, the difference will be that the product, if you want to develop this kind of product, you will have to apply different regulation or different guidelines, because the regulation for advanced therapy medicinal product is somehow different from the legislation for other biologics. So this is why it is important to keep in mind that the classification is very important for the development of medicinal products. And the second point I would like to discuss with you is the point regarding orphan similarity, because I'm sure you are all aware of what is called the orphan drug designation procedure, which may allow a pharmaceutical company to protect the product once it is on the market, to protect the product from competitors for 8 or 10 years. And if there is a competitor, you will have to demonstrate a significant benefit if you want to come to the market. This was initially intended to foster research and development for rare diseases. But the drawback of this is that in some cases it can block improvement in what can be done with gene editing or gene therapy. And I will just give you just an example. This is just the Commission regulation, which is at present the regulation. And according to this regulation, we will have to look, just to compare two products, at the active substance of the product, looking at if the active substance is similar or not. And there are also very complicated definition of what is a similar active substance, which means an identical active substance or an active substance with the same principal molecular structural features, but not necessarily all the same molecular structural features, and which acts via the same mechanism. What is really worrying at the moment is that if you look at the present regulation, two related cell therapy products are considered similar, but you don't know what are related cell therapy products. And if you look at gene therapy, two polynucleotide substances, when the differences are in the vector or the transfer system, are considered as similar. So just to give you an example, you know that by gene therapy, you can modify cells by adding a gene into the cell. This is what is called additive gene therapy, and we have seen a lot of examples of this today. If now you look at the left part of this diagram, this is Stringvelis. Stringvelis is the product which has been granted marketing authorization, is made of CD34 positive cells that have been transduced with a retroviral vector carrying the ADA gene, the ADA sequence that will make the expression of the ADA protein that will treat the patient. If you look at the second drawing, if you have CD34 positive cells that are transduced with a lentiviral vector, which is not the same vector, but then the same vector will carry the same therapeutic sequence, because the therapeutic sequence is the expression of the ADA gene, and the expression of this gene will synthesize the ADA protein. So just now, these two products are considered as similar, because the difference is not in the sequence, but in the vector system. If you now look at gene editing, now you want to edit the gene into these CD34 positive cells. If you are able to correct the mutation, then the therapeutic sequence will be the expression of the normal ADA gene. And then if you look at the polynucleotide sequence, the polynucleotide sequence will be the same in all these different cells. So because Stringvelis is now on the market, these two other products should not be allowed to go to the market before 8 or 10 years. Which means that there is a way of blocking improvement, because if you consider that lentiviral vector is better than a retroviral vector, or if you consider that gene editing is an improvement as compared to retroviral vector, you will have to demonstrate a significant benefit. And because ADA is a very rare disease, you won't be able to have enough patients to make a comparative study to compare gene editing with retroviral vector. So this is why it's important today that the legislation be modified. And of course, there is at the present a public document that has been issued for consultation until today. And of course, the aim of this document is to modify the present regulation just with new definition. I don't go directly into these definitions, but just to make sure that if you have two gene therapy medicinal products, when the differences are in the vector, then you can consider now that the two products are different. And then, of course, the protection of the orphan drug designation will not block any improvement for new products. So in a certain way, it's a double-edged sword, because some companies will say that if they are no longer protected, they will no longer be interested in developing very specific product for very specific diseases, because if they don't have protection after marketing authorization, they will not have any enough return on investment to develop the product. So this is why I wanted to show you that the definition is important, because if it's no longer a gene therapy medicinal product, of course, theoretically, the orphan similarity is no longer an issue, because it's two different types of product. One is gene therapy, the other one is biology. So the classification is very important. And then, of course, also, the definition of orphan drug designation is also very important. This is why I think it is important that these two guys, Michele and Chris, continue to defend the position of the patient and the cat, because the classification is not only an issue, a theoretical issue or a regulatory issue, but I think it is very important also for the patient to have this participation of patient to the different committees. And I thank you for your attention.